Hi students, today's video lecture is going to continue with meiosis and we're going to specifically focus on how meiosis, which is a type of cell division that produces gametes, egg and sperm, also produces gametes that are diverse genetically. And the way that I explain this for you to think about is think of two parents that have 5 kids or 4 kids. It's always the same, not the same, but it's always eggs from the mother, sperm from the father that fuse to produce these kids. But these kids aren't identical. They're all different. So somehow each of the egg that the mother produces and the sperm that the father produces are different than the other egg and sperm. And no matter how many kids a pair would have, they would all be genetically diverse. And so for today's lecture, I want you to focus on what parts of meiosis produce this genetic variation. Okay, and this is section 10.2 in your book. Again, I kind of have abbreviated it. So we talked about meiosis during the last video lecture, which is similar to mitosis, except that it has 2 divisions. It has meiosis 1 and meiosis 2. Meiosis 1 produces 2 cells, and then in meiosis 2, those 2 cells divide again to form 4. And the part of this cell division that produces genetic variation all occurs in meiosis 1. All right, meiosis 1 is very important. It's the one that is referred to as the reduction division. So those homologous chromosomes that we've discussed split, right? And each cell only has one of the homologous pairs. So in humans, in the beginning of meiosis 1, we have 46 chromosomes or 23 pairs. By the end of meiosis 1, we have 2 cells, each with only 23 chromosomes and no pairs. All right. So there's 2 ways that meiosis 1 produces genetic variation. One that we'll talk about is referred to as crossing over, and the second is independent assortment. And both of these will occur in metaphase of meiosis 1. But before we get that, I want to tell you something else that's unique about meiosis compared to mitosis. So this picture I have here is of prophase 1. So the prophase in the first division. And if you remember, prophase is when that chromatin condenses into visible chromosomes. We see the chromosomes here in this picture. All right, these chromosomes are duplicated because they have 2 sister chromatids. And in meiosis 1, these homologous pairs during prophase 1 find each other. This does not occur in prophase of mitosis. So in prophase 1, these homologous pairs, remember they're homologous pairs because they are of the same size, they contain the same genes, but those genes can be in different forms and they find each other. And that is referred to as synapses. Synapsis right here, synapsis, is when chromosomes, homologous chromosomes find each other. That got cut off here in my notes. But synapsis occurs in prophase 1 and when these homologous pairs find each other. So you can see up here, these are homologous chromosomes, and down here, they're homologous chromosomes because they are of the same size, their centromeres in the same position, they contain the same genes. Okay, so after that, what happens after prophase? Remember, PMAT, prophase, metaphase. They line up in the middle. And when they line up in the middle, and they're all close to each other, the homologous pair, something that's called crossing over occurs. And we're going to do this in lab, where you can see here, start in this picture, what they're trying to show you are 2 homologous chromosomes. The red represents the sister chromatids of 1 duplicated chromosome. The blue represents the sister chromatids, the 2 sister chromatids of the other homologous chromosome. And these chromatids kind of overlap with each other. So the chromatid from the first chromosome, do you see, is kind of looping over a chromatid from the second chromosome, duplicated chromosome. So you see that the red chromatid is kind of overlapping with the blue, and they kind of swap pieces. They cross over, literally, and they kind of break off, and this part will end up with this one. I guess the picture shows it better. And so this crossing over now, look on the right, it's producing chromosomes, duplicated chromosomes that have parts from the other chromosome. Okay, so it's an exchange of genetic material between non-sister chromatids. The reason they say non-sister chromatids is because sister chromatids are from one duplicated chromosome. So here the exchange is happening between a sister chromatid from one duplicated chromatid and a sister chromatid from another duplicated chromosome. So you end up with this new combination of genes, right, along the chromosome. And that occurs during... Okay, I want to clarify one thing that, depending on the source that you read, may say two different things. The crossing over that occurs after synapses. So when prophase synapses happens, where the homologous pairs of chromosomes find each other. And then the exact point in which crossing over occurs can vary between sometimes happening in prophase and sometimes happening in metaphase. And so earlier I say metaphase, but in lab and in your textbook, they refer to it occurring in prophase. So synapsis occurs in prophase. The homologous pairs find each other. And if you look at this picture, it looks like the crossing over is actually happening in metaphase. So that's why I'm bringing this up. So synapsis, prophase, crossing over, and independent assortment. Independent assortment has to happen in metaphase. I just want to make clear that crossing over occurs kind of throughout prophase into metaphase. Anaphase, they separate, right? And this crossing, this independent assortment just means that the homologous pairs can line up either with the paternal on the right, maternal on the left, or vice versa. And each homologous pair assorts itself independently of the other. So each has a 50-50 chance to be either on the left or on the right. Anaphase, the homologous pairs separate. Telophase, right, they start to unravel, the nuclear envelope starts to reform, and by the end of meiosis 1, we have 2 cells with half the number of chromosomes, so now we're back to haploid, right? And there's still duplicated chromosomes, so that's why we need the second division of meiosis. So, independent assortment occurs during metaphase, remember meta, middle, and meiosis, the homologous pairs are going to line up in the metaphase plate. In mitosis, it would have only been like one duplicated chromosome lining up. In meiosis, it's the pair that line up. Because recall, the point of meiosis 1 is to separate those pairs. And when they line up, when homologous chromosomes pairs align at the metaphase plate, they align in random order, or they sort themselves independently. And here's what I mean by that. Let's look at this cell we have here. How many homologous pairs of chromosomes does this cell have? Yes, this cell has 3 homologous pairs. And each of those homologous pairs can either line up with the blue on the left, as you see it now, and the red on the right, or they could also line up with the red chromosome on the right, the left, and the blue chromosome on the right, right? So if this was happening in gametes from your body, the blue represents the chromosomes you inherited from your father, the red represent the chromosomes you inherited from the mother. So they can line up in any of those ways. They align themselves, each pair align themselves independently of the others. That's why it's referred to as independent assortment. And so this is one combination. Here, what's the difference? Look at the last homologous pair on the bottom. See how the red one lined up on the left and the blue one on the right? In combination 3, the middle pair flipped. In combination 4, the top flipped. And so there are 8 different combinations by which these 3 homologous pairs can line up. So we would say that in this case, it's 2 to the 3rd power, because there's 2 ways each of those 3 homologous pairs can line up. So that's 8 different combinations, which are all here. Humans have 23 pairs of homologous chromosomes. So that would be 2 to the 23rd power combinations, and that would be thousands of different combinations. I think it comes out to like 8,000 something. And so that's why every egg that a mother makes, because of independent assortment, and then on top of that crossing over, are all genetically different. And the same goes with the male. All right, I hope that made sense. All right, so that's meiosis 1. Meiosis 1 produces 2 cells. If we start in humans with 46 chromosomes, after meiosis 1, we've separated the homologous pairs. So we have in each of the 2 cells 23 chromosomes that don't have a homologous pair, right? So the first division is referred to as the reduction division, and that's when all the genetic variation occurs. The second meiosis, meiosis 2, is exactly the same as mitosis, except we're starting with 23 chromosomes instead of 46, but we also end with 23 chromosomes. So meiosis 2, I'm not going to spend a bunch of time on, but we start, look up here, with 2 cells, right? And then from there, prophase, they condense, right? They become visible. Metaphase, they line up along the metaphase plate. And now the point of meiosis 2 is just to separate those sister chromatids, just as in mitosis, because these chromosomes are duplicated. And at the end, we don't need the chromosomes to be duplicated. So anaphase, the sister chromatids separate in each of the cells. Telophase, they reach to the poles. The nuclear envelope starts to reform. The chromosomes start to unravel back into chromatin, right? And cytokinesis is starting. And by the end, you have 1, 2, 3, 4 daughter cells, each with 23, not in humans, it would be 23 non-duplicated chromosomes. And this is just a little summary of meiosis compared to mitosis. So meiosis is 2 divisions. Meiosis 1, the reduction division, where we separate the homologous pairs, so we cut the chromosomes number in half. Mitosis is only 1 division. Meiosis has synapses during prophase, where those homologous pairs find each other. That does not occur in mitosis. In metaphase, there's crossing over in meiosis. That does not occur in mitosis. Meiosis 1 halves, right, the chromosome number. Mitosis does not do that. Meiosis produces daughter cells that are genetically different from the parent and from each other. Mitosis produces always identical cells. That's the point. We are just dividing but making clones. Meiosis is for sexual reproduction, produces gametes, egg, and sperm. All right, that is it for this explanation of meiosis. That finishes meiosis. Next, we will move on to genetics. Take care, everyone.